numbers on the right-hand side. Okay, we will.
When I think about cloning, I think of how cartoons portray it, where someone pushes a button and then they transfer what's in container A to container B, as so. They push the button and they copy it to container B. Well, this technically isn't cloning in society because we can't do that. We haven't developed that technology yet. But everyone can see twins out on the street. Twins necessarily aren't clones because they aren't identical. They don't have the same makeup, they don't have like the same genes or anything. They do have similar genes but not the same genes. They have fingerprints. Their fingerprints aren't the same either. Being a twin myself, I'm very into cloning. I want to research cloning because I want to see what it is. I've researched cloning over the past few weeks to explain to inform to you how cloning works. Today we will learn about the history and basics of cloning and where the word cloning came from, and the types of cloning, how they are made in labs, and the different types in nature. And then we're going to focus on Dog the sheep and why she's important to cloning. The first one is cloning. Cloning is the process of taking genetic information from one living thing and create identical copies of it. This is stated as according to Elena Bradford's published article, Facts About Cloning on March 1st, 2017. She is credible because she has spent 16 years covering health research and has many certifications at Oklahoma State University. Now the Greek root meaning is twin, or breaking in two, stated from, according to Biotechnology's article, The Cloning of Dolly and Other Mammals, published October 9, 2012. They're credible because they have Indiana University biology faculty who review the articles, and they all have PhDs in their field of science. Now, when I mean twin, it's not like the twins we see on the street today. Again, they don't have the same fingerprints, so therefore they're not technically clones since clones are identical. So now we're going to talk about the history of cloning. The first person who discovered cloning was Hans Adolf Edward Dreyer in 1885. He discovered if you take a sea urchin, or a two-celled embryo from a sea urchin, and you shake it really hard, those two cells in that embryo will come apart, and they will then grow into their own sea urchin. Hans Spemann discovered in 1902 that if you take a salamander, two-celled embryo from a salamander, and you tie a hair around it, and you pull it really tight, it can pull those cells apart too. This is credible by the Utah University article on cloning published July 10, 2014. They're credible because they have faculty who have PhDs in their field of study. And that is a two-celled embryo. Now we're going to go on to the different types of cloning. The first one, naturally in nature. It's asexual reproduction. This is the offspring is produced from a single parent. This happens in mold and bacteria, and the offspring has the same identical makeup as the parent. The next one in the laboratories is gene cloning. DNA is extracted and enzymes are used to break the bonds between nucleotides, and then the broken DNA is then fused with a plasmid, resulting in a cloned DNA strand. A plasmid is a self-replicating gene element. This type of cloning is used only for genes. You don't create a whole thing. This helps scientists learn how genes are, their function of genes and how they work, and how it makes a person who they are, or organism who they are. The next type is therapeutic cloning. Transferring a diploid nucleus from a body cell into an egg whose nucleus has been removed, and the two fetuses together creating an egg. This is most common when you have a burn or you have severe flesh that has been removed. They use this so they can replicate your cells and make it heal faster. This is credible by according to the U.S. National Library of Medicine's article about DNA, published September 12, 2017. They're credible because they have research done by faculty that have a master's degree or above in their field of study. And the main one today, we're going to talk about reproductive cloning. Reproductive cloning is a process created by an organism in a laboratory by extracting DNA from an organism and placing it to an egg of another organism that has the DNA to be birthed. Now this is a picture of reproductive cloning. You take the nucleus, which is a DNA, out of a cell, and you take an egg from this sheet. And then you combine them together, but you take the DNA out of this one and put it in that one. And therefore, you put that one in the sheet that the egg cell came from, and then they can birth a lamb. Various animals have been cloned by this, like rabbits, cows, sheep, cats, dogs, and it is the most common way to clone animals. And this is how Dolly the sheep was cloned. Now, Dolly the sheep. Dolly was the first mammal to be cloned from an adult cell, non-embryo. All the other animals that were produced from reproductive cloning were from embryos. Dolly was made from, her cell that she was produced from came from a 6-year-old sheep udder. Dolly was one out of 277 attempts. She was born July 5th, 1996 in the Rosen Institute of the United Kingdom. She suffered from arthritis and pulmonary adenomatosis, which is a lung tumor that happens in sheep that are raised indoors. Now, Dolly was euthanized on February 14, 2003. Now that she was made from a 6-year-old sheep, sheep can only live from 6 to 12 years old. And since she was made from a 6-year-old sheep, she only lived to be 6 years old before she couldn't walk anymore. This is credible by Carl Zimmer, who published What Dolly Wrought. Retro Report looks at Dolly on October 14, 2013. It's credible because he has numerous books, articles, and publications with the National Geography, which have won awards for his research and writing about nature and its biology. To sum up, we have learned the history and basics, how Hans Dreich and Hans Spemann discovered cloning, the different types of clonings and how they are produced, and we know how the impact Dolly had on cloning and why she was important to science.